Hello biology family. Today we will discuss an important technique in molecular biology, which is polymerase chain reaction or PCR. I am Dr. H and don't forget to subscribe my channel. So PCR was first invented by Carey Mullis in 1989. He also received Nobel Prize for this noble invention in 1993. PCR is basically a method of amplifying the target sequence of DNA. And by this technique, millions of copies of DNA can be produced within few hours with higher sensitivity and selectivity. This technique is very rapid and PCR has ability to amplify DNA sequences as short as 50 to 100 base pair and as long as 10 kb. To start a PCR, certain things are required that can amplify any target sequence. These are number one, a known sequence of gene of interest. Number two DNA primers forward and reverse, and each primer should be of 20 to 30 nucleotides. Number three, Taq polymerase, which is required for synthesis of new DNA strand. This enzyme is basically derived from bacteria Thermus aquaticus that are found in hot springs. Therefore, this enzyme is not denatured at high temperature. And lastly, the DNTPs, which are all 4-deoxyribonucleotide triphosphates. Once you have all these things, you can carry out a PCR through a thermocycler or also known as a PCR machine. Now I will show you how exactly a target sequence is amplified in different steps. You can see there is a double-stranded DNA with a region to be amplified. This DNA sample is first heated during denaturation with a melting temperature of 95 degrees Celsius for about 15 seconds to 2 minutes, and as a result, the strands are separated. Then, primers are added that are annealed to separated strands by cooling to 50 degrees Celsius for at least 1 second to 2 minutes. The new strands are then synthesized by the Taq polymerase in presence of all 4 DNTPs. Polymerase chain reaction is allowed to take place at 72 degrees Celsius for 30 seconds. This is the first cycle of PCR. The two DNA strands each serve as a template for synthesis of new DNA from the two primers. Now that we have 4 DNA strands from the initial 2 strands, the same steps are repeated in cycle 2. Strands are separated by denaturation, but this time there are 4 strands. Again, new strands are synthesized in each cycle. DNA strands are doubled. PCR is now being a common technique and is used for many purposes. It is used to detect infectious agents, especially latent viruses and bacteria. Identifying single gene disorders, for example, cystic fibrosis, thalassemia, sickle cell anemia, and many other chromosomal abnormalities. PCR's exponential amplification enables genetic analysis even from tiny, old, degraded DNA in archaeological samples, which allows the study of ancestry and evolution. PCR enables early cancer detection by amplifying trace amounts of tumor-specific DNA mutations or abnormal gene expressions from blood, tissue, or other body fluids. PCR also allows the DNA in a single cell, hair follicle, or spermatozoa to be amplified and analyzed. Thus, the application of PCR to forensic medicines is very obvious. There are many different types of PCR. In the next video, we will discuss these types. So stay tuned for more amazing techniques of molecular biology.